What do you mean when you say that there is no evidence of the coronavirus causing disease?
What I've done is I've gone ahead and looked at all of the papers where there are scientists claiming to find evidence for a new virus, that they claim to isolate the virus. And I've basically scrutinized their procedures, and I've compared that to the history of how viruses and other diseases are proven to cause disease and how they're identified. So I've become familiar with the procedures and how they work. And what I've found is that the scientific procedures that have been utilized in all of these scientific studies are actually, it wouldn't be possible using those techniques to isolate a virus and purify it and prove that it exists. So what they've done really is they've done kind of two things. So one has to do with the genetic material, and one has to do with isolating a viral particle. So I'll talk about those separately. I'll talk first about the genetic material. So the general procedure that they utilize is that they identify someone with an illness that is unexplained, like this happened with the first group of patients in Wuhan, China, but it's been done in several other places around the world. And they have taken lung fluid from those patients, and they do this through an invasive procedure where they put a fiber optic scope through the mouth or nose down in through the windpipe into the lungs, and they squirt some fluid there and slosh it around, and then they suck it back up. And that would have basically a mixture of different things. It's a very impure sample, so it would contain, secretions from that area, like sputum. And it would also contain a mixture of different cells, such as our own lung cells and our immune cells. It would also contain various microorganisms that are present in our normal tissues, like bacteria and fungi. And then it also, lung fluid has shown to contain free genetic material as well. And then it would also contain what's called exosomes, which have their own genetic material, and I'll elaborate on what they are in just a minute. And so they took this sample of lung fluid, which has genetic material from many sources, and they added an enzyme that would digest any cell membrane, so it basically allows the DNA or RNA to be free in the solution so that they could do further analysis. And then they've identified a sequence of RNA that they allege is from a coronavirus or from the coronavirus family. But the thing is that since this sample is impure and has many sources of RNA, it's impossible to know what the origin of it is. If they wanted to be certain that it came from a virus, what they would do is take that lung fluid and filter it, because you can purify viral particles by filtration because they're much smaller than any other cells, even bacteria cells. And then you typically use a centrifuge with a density gradient that creates a tight band of these particles that's easy to suck up in a pipette. And then you could take those particles and extract the genetic material and say that it came from those particles. But when you have an impure sample with lots of sources of genetic material, you really don't know where it came from. So what their solution to this problem is to compare the identity of the sequence of this RNA with other known RNA sequences. And they specifically compared it with the SARS-CoV-1 virus, which was the virus allegedly responsible for the SARS outbreak in 2003.
You say allegedly. We'll get to that, I guess.
Yeah, well, okay. The thing is that they used the same exact procedures when they did the experiments for that virus. So, in other words, they got the genetic material from an impure sample also, so it's not clear where the origin of that is. So you can follow this back in a chain to some of the original papers, and what I've found pursuing that is essentially the same exact procedures used each time. But how they said that this represented a coronavirus and was related to the SARS-CoV-1 virus, in fact, they call this virus the SARS-CoV-2, is that they looked at the identical nature of the sequences, and they found that it was just under 80% identical. And that's how they said, well, it must be related, and so we'll call it SARS-CoV-2. But the thing is, if you look at the genetic sequence between humans and chimpanzees, you'll find that there's 96% sequence homology or identicality. And so we are clearly not the same as chimpanzees, and no one would say that our genetic material is part of the chimpanzee family. But yet they're using a sequence identity that has a much lower percentage, under 80% compared to 96, and saying that's a coronavirus because of that. So in my opinion, this is extremely weak science, and without purifying the lung fluid sample, you really can't tell what the origin of the RNA is. So they've used this sequence to develop the main diagnostic test that they're using, which is the PCR test. And aside from any problems using the PCR methodology, we simply don't know what it's testing for because we don't know the source of that genetic material.
Okay, so they're going around testing people and they're getting back these results, and the results don't necessarily correlate to the person they're testing having anything that might be related to something that might cause a disease. You could get a quote-unquote false positive for other things in the past, including maybe flu vaccines or colds or a variety of things that happened in your past, and now they're qualifying you as COVID-19 positive. Is that right?
Yeah, well, it's quite possible that this RNA actually comes from our own cells. So a positive test could just say that we've measured your own RNA. I mean, the thing is, we really don't know what it is, and it doesn't seem to correlate with presenting with actual clinical illness. So lots of people who don't have any symptoms and didn't get ill test positive for this, as well as many people who are ill and have symptoms test negative. So since we don't really know what we're testing, in my opinion, we can't trust the results of this test at all.
I wanted to ask you quickly about the exosomes, because this is something that David Icke mentioned and specifically gave you kind of the credit for it. Maybe you could just define what an exosome is to people, maybe talk about what they are, and can some of these kind of false positives be induced by stress and fear and trauma, which is going on in droves right now?
Yeah, absolutely. So what exosomes are, and they were discovered by scientists about 30 years ago, is that they are small vesicles or little sacs of fluid that bud out of our own cells. So they're secreted by our own cells. Every cell type in our body and every cell of every mammal at least makes exosomes. And they're made at a baseline rate all the time. So you're making them right now, as am I. But when you have some kind of illness, and a variety of different illnesses have been shown to cause this, it causes your body to induce exosomes and produce more of them. And the reasons for that are thought to be, the main function is that it's thought that they play is in communicating between different cells that are far apart in your body. So it's not the neighbor that they can communicate with other ways, but some remote part of your body. So the structure of exosomes would be that it's very simple, they're very small, the same size as virus particles. There's a range of sizes, but there are for viruses as well. And they have a very simple structure, just a membrane and maybe one or two proteins on the membrane. Usually one of them is a receptor that is like a key mechanism that finds the right lock on the target cell so it can go into the target cell where it's supposed to communicate. And then inside the vesicle is some kind of genetic material. And it could be a variety of different materials, including various forms of DNA and RNA, just as it has been described in virus particles. So there's another role that has been demonstrated in scientific studies where sometimes these exosomes can actually take up toxic materials like bacterial endotoxins from the environment outside the cells, and then prevent the cells from being exposed to those toxic materials. So that may be one role. So the reason why this is so important is, one, because the similarities have been recognized by scientists, including virologists, and many times scientists have actually said they're the same thing or they have substantial overlap, including prominent virologists. And then they've also found them to have sometimes a combination of what they call viral RNA or DNA mixed with human RNA or DNA. So remember from what I said earlier that we really don't know the true source of what they call viral genetic material, but nonetheless, whatever it is, they found it in our own body, in these exosomes. So the procedure that's used to isolate and identify a new virus, including the SARS-CoV-2, which is the virus associated with COVID-19, what they do is they take the lung fluid from a sick person, and instead of just trying to purify a virus particle out of that, they mix it with a foreign cell. And most commonly, they use vero cells, which are derived from the kidneys of monkeys. And then they add antibiotics to the cells as well. Now, there are several studies that show that antibiotics is actually one thing that can induce exosomes because it's generally toxic to the cells. So they're basically created a recipe where they're mixing the lung fluid with cells and antibiotics that are going to make exosomes. Right? There's no two way about it. They're going to be exosomes from those cells when you mix them with antibiotics. So they then, grow these cells for a few days, up to a week or two, and then they look at it under the microscope, and they see little particles and say that that's the virus. But the thing is that they're going to be exosomes in the mixture because you've created the recipe to make exosomes, and they don't account for them at all. So they don't have like a photograph where there's, a red arrow that points to something they say is a virus, and then a blue arrow that points to something that's an exosome, and they show how they're different from each other. And then they would also then have to purify those from each other to further characterize what's in the virus or the exosome. But they don't mention this at all. So the only conclusion I can come to is that what we're seeing in the pictures must be exosomes because they're going to be there because they basically created the recipe to make exosomes, and they don't account for them in any other way. So that has to be what we're looking at.
All right, and so this can also cause these false positive results, and can they also be related to stress?
Well, the test is not testing for a virus, and it's not testing for an exosome. It's testing for a specific genetic sequence. And we don't really know where that genetic sequence comes from, so we don't really know what the test is testing for. But the way that you would design such a test is that you would develop what's called a gold standard. And this is also something that I was taught about in medical school, and they did actually give us an example of this, and it's to diagnose a pulmonary embolism, which is a blood clot in the lungs. And the gold standard test is what's known as an angiogram, where they inject dye into your blood vessels, and then they show under a special type of video X-ray that the dye stops because something's blocking it. And that's the blood clot. Now, and they compared that with another test called a VQ scan, or a ventilation perfusion scan. And they compare this new test, the scan, to the gold standard. And that's how they determine, is it accurate? Does it capture every case? Does it give you false positives? What's the error rate? Now, for COVID-19, they've never demonstrated that they have purified and isolated a specific viral particle, but that would be a gold standard. So if, let's say, they took 100 patients, and from 100 patients, they isolated this viral particle that they could easily identify as to what it was. And then they compared that result to the results of a PCR test or an antibody test or any other test they wanted to develop. They would then be able to see how accurate it was and calculate an error rate. But this has never been done because there's no gold standard test. They're trying to say that this genetic test is the gold standard, but there's no way it could be because we don't really know the source of what it's testing. There was one study that tried to estimate the accuracy of the test, and it did this by using some mathematical analysis, like statistical analysis, not a computer model. And then it also followed people longitudinally. So they would have a test result, they'd be asymptomatic, and then you wait a couple of weeks and say, do they get sick or not? And they estimated the false positive rate of the test to be 80%. So in other words, 4 out of 5 people that have a positive test don't have any illness. So very, very inaccurate testing. We don't know what it measures. It has no known error rate. So basically, essentially, it's just worthless. But the exosomes is what they're showing under the microscope and saying is a virus. And that's really where the exosome becomes most relevant. Now, whenever we're sick from virtually any type of illness, and even from psychological shock. So if we, suddenly lose a loved one to a traffic accident or something like that, we would actually make more exosomes due to the response that we have, the psychological response. And they've been shown to occur with toxins like heavy metals or pesticides or antibiotics. They're also shown to be induced with ionizing radiation, like X-rays or gamma rays, with infections, with asthma. So pretty much almost any illness will cause us to make more exosomes, and they're a response to the underlying disease state and most likely function to help us recover from that. You could think about them as sort of a collaborator or a partner with our immune system in that respect.